The overall goal of this procedure is to create an inexpensive and effective filter for concentrating a wide variety of waterborne pathogens from large volumes of water. Filter construction is achieved by first rinsing glass wool, washing it with both an acid and a base, and re-rinsing the wool to a neutral pH. Next, the glass wool is rinsed in phosphate-buffered saline. In the next step, the glass wool is packed into a PVC pipe to form the glass wool filter. After using the filter for sampling water, an elution step is performed. The sample can then be stored, concentrated further, or proceed to be analyzed for pathogens by cultural or molecular methods. Waterborne disease is a significant threat to public health worldwide. Disease transmission can result from drinking water and recreational water activities. One approach for evaluating the sanitary quality of water is to test it directly for disease-causing pathogens. There are several benefits for using glass wool filters as opposed to other traditional waterborne pathogen filters. First, the filters are inexpensive, allowing for a large number of samples. Second, glass wool allows the concentration of a large volume of water, which is often necessary for detecting waterborne pathogens. Third, glass wool filters are also very portable, making it easy to conduct field sampling in remote locations. Glass wool filters are capable of concentrating a variety of pathogens, including viruses, bacteria, and protozoa. Finally, used in conjunction with a pre-filter, glass wool filters will concentrate pathogens in difficult water matrices. The materials required for making glass wool filters are safety equipment,